on, but I'd like to introduce Dr. Will Rickey. He will be introducing Dr. Wessells for today's talk. And Dr. Rickey probably doesn't need much of an introduction to this audience, but he is, as you know, the PI of the UW-Madison O'Brien Center, the Urology O'Brien Center. He's got many other funded grants under his belt from NIH and other organizations. And he's in his second term of this O'Brien Center grant, which is a very successful and leading O'Brien Center in the field of benign prostate research, as well as in CAIRIBU. So Dr. Rickey, I would like to introduce you to introduce Dr. Wessells, and then we'll get started.
Thank you, Chris, and thank you for the invite to moderate and introduce this inaugural event, one I hope we see many more of, and based on the audience, it looks like there's a great participation ready to be had. I'm certainly happy to introduce Dr. Hunter Wessells. Hunter got his BS and MD from Georgetown University. His success has led him to become, in 2008, the professor and Nelson Chair of Urology at the University of Washington School of Medicine. He specializes clinically and research-wise in genital urinary trauma, reconstructive surgery, Stone's disease, erectile dysfunction, and many other things regarding the urology. Additionally, he is passionate about training our next generation of physician scientists, and personally speaking, he's training one of our past MD PhDs, Tristan Nicholson, and we've been very impressed of what she's done under his tutelage. He's been and continues to be funded by a number of agencies, including NIH and NASA, and he serves a number of roles in leadership in the urologic field, including the AUA Guidelines Panel Chair for male urethral stricture and other urologic field roles. On a more personal note, he's an avid outdoorsman, sportsman, enjoys open water swimming, backcountry skiing, tennis, and he's a farmer at heart. He enjoys organic vegetable gardening, and perhaps more importantly, from my viewpoint, the intricacies of winemaking, especially Burgundy wines. And on that note, Hunter, I'd like to offer up a potential collaborative invite. If you make the wine, I'll taste it, and we can get it out the door. Today, his talk is entitled Comprehensive ED Phenotyping and the Urologic Complications of Diabetes Studies, or UroEDIC, and beyond. So with that, I'll turn it over to Hunter. Thank you, Hunter.
Thanks, Rick and Chris and the whole group. I'm sure some people could maybe mute their microphones while I'm talking. Thanks. It's really great to be with the University of Wisconsin, the other UW, that's what we all call each other, and to share with you something that I've been working on for nearly 20 years. So I'm going to try and share a story of how I got to be involved in this project, and hopefully whether you're a trainee or a starting investigator or a senior investigator, there's something that I can share with you that'll be valuable. And the bottom line is, if you learn to do something really well, you can apply it to many different areas. And I've been trying to learn how to phenotype erectile dysfunction. So with that, I'm going to get started on the story. And what we've decided is that you are not going to interrupt me along the way, which is how we do it at the University of Washington. And you're going to actually save your questions till the end and use the chat or put the questions to Chris in some other way, and then we'll go over them at the end. So I'm going to start by just sharing this timeline of this landmark study called the Diabetes Control and Complications Trial, and its observational follow-up, which is called the Epidemiology of Diabetes Interventions and Complications, and how we got involved in the urologic complication phenotyping, and how this has been a really rewarding line of investigation that we hope will continue to answer questions. It was commissioned back in 1978. There was an RFA. There was a planning and feasibility section for this trial to make sure that they could do what they wanted to do. And then they ran the trial for 10 years from 83 to 93. And the amazing thing is then since 1994, for the last 27 years, almost the entire cohort of 1,642 men and women have been in an observational follow-up. And the things that have been learned subsequently span the entire field of medicine: heart disease, blindness, you name it, it's been studied. And we were fortunate to get involved in 2003, and it seemed like we were late to the game at that point because so much time had already passed. But we've been actually working for 18 years now, and in 2010 we got funding to rephenotype the cohort and then to have some sort of assessment every year. And so now we're going on having 10 years of repeated measures on this cohort, which I think offers a lot of powerful tools to study causation and move from just describing or just looking at a cross section to actually seeing the development of complications over time. This is just a basic outline of the trial, which will become relevant. So actually 1,441, I misspoke the number there. And these men and women were randomized to either conventional treatment, which at the time was 2 doses of insulin a day, and just make sure you don't get hypoglycemic, or intensive therapy, which was 4 shots a day with finger sticks and measuring blood glucose or using a pump. And we've followed these patients over time, and you can see that we always had close to 1,200 patients in UroEDIC. What's interesting, we actually got more people with our second iteration because these men and women actually were interested in what was going to happen to them and their urologic complications. And so they wanted to participate in UroEDIC. And we've just been funded again for UroEDIC 3, which is going to be focusing on bladder dysfunction over the next 4 years. So that's really exciting. A long gap between funding, we weren't funded for 10 or 11 years, we were funded for 5, but we've managed to keep the team together and be prepared for the next phase. So a couple of the just big picture findings, and then I'm going to move into the phenotyping. These are the different urologic conditions that we saw in the women in the study. And you can see that about a third of women had no urologic complications, and then progressively fewer had more and more. But so there are about a third of the participants who have, multiple urologic complications. The most common being incontinence, and these Venn diagrams are not to scale. For men, the picture was very similar. The percentages across the bottom are very similar, and erectile dysfunction was the most common disorder self-reported in the men. So just a quick take home right away is that, leveraging these existing cohorts can be really powerful for a number of reasons. For some of our benign urology conditions, no one is going to stand up a cohort of 1,400 patients. They just won't have the money to do it, and they won't see our conditions as important enough to invest in to this degree. So we have to collaborate and piggyback. And so for diabetes, for type 1 diabetes, this was a great study. But it's still, the participants are now aging, and now they're in their 50s and 60s. So we can't keep studying them forever. We have to find other cohorts. And there's something called the type 1 diabetes exchange, which has, a much larger group of potential participants. And it's something that people studying type 1 complications should look into. And then there are a number of type 2 cohorts that might be available either for secondary data analysis or biospecimen use, and those are important. I think what we learned from UroEDIC was that focusing on the urologic symptoms actually enhanced participant retention and engagement within the parent study. And if you're working with another parent study, you can cite us for that. And a cool thing is that now our questions are embedded into the parent protocol. So we don't have to go back every couple of years and beg for them to keep our urology questions in the the study manual, that was always a challenge. Well, we only get X number of questions, but by embedding them in the parent study, A, it costs less, and B, we continue to get the data every year. And I mean, clearly it takes perseverance and collaboration to do this. And we started in 2003. It took a long time to get funded for the first time. It's almost, we're on like a 10-year cycle to get funded, but it's been really valuable. And a point I'd like to share is that biospecimen use amongst these studies is really important to NIH, and I think it's something that's underutilized. So if you can propose an ancillary study where you're going to use existing biospecimens, that's going to reinforce value to the institution. I'd now like to shift gears and talk a little bit about ED phenotyping, and we'll show you what we did in UroEDIC, and then I'll also show you how we're trying to apply that to other cohorts and other problems. Back when the DCCT was created in 1983, pretty much people were just using the term impotence, and there were no measures that had been developed to measure it. So they actually had a yes/no question from the inception of DCCT. Did you have impotence this year, yes or no? NIH consensus conference in 1989 made it clear that we should use the term erectile dysfunction. And shortly after that, a number of studies, including the National Health and Social Life Survey study and the Massachusetts Male Aging study, used better questions. In 92, it was a dichotomous response, and in 94, it was self-reported response to the question, can you get and keep an erection sufficient for sexual activity? The prostate cancer prevention trial used that same question as MMAS, and they actually repeated it yearly for 7 years. And that data has never been fully tapped, but it's a large cohort of men. The NHANES, the National Health and Nutritional Exam and Survey, had an ED question, the same one for 1 year. So we got some nice nationally representative data, but it was only a single time point. A study in Australia gave the IIEF, the International Index of Erectile Function, 5 years apart. So they got some interesting data on remission and progression of disease. And then we've been using the IIEF at certain time intervals. And then we have a 5-item single question, which has been given annually since 2010. So this is a little bit of the evolution of ED phenotyping. I mean, when possible, yes, we'd like to use the whole IIEF questionnaire, but that's 15 questions and it may be burdensome. So we narrowed it down to the erectile function domain, and sometimes, some years we just used the single question that had 5 items. And I think there's a lot of literature to suggest that you don't need a whole battery for certain phenotypes. So that's an important lesson, I think, that you have to be flexible. And if you say, well, I want my 50 items in your ancillary study, they may just say no. And you'll have to really do your research and find out what questions do you really need. So this is just some data from UroEDIC, back in 2003, and it highlights, I think, some of the questions for us. So this is the frequency of male participants in the study by their hemoglobin A1C, and you can see, the target is to have a hemoglobin A1C lower than 7, but in type 1 diabetes, that's just not possible for a lot of patients. And here you can see the distribution of individuals. And what's amazing is that you can have someone out here with an A1C of 11.2 who did not receive the really intensive treatment during the randomized period. They smoke and they still don't have erectile dysfunction. And then you can have someone down at the other end of the spectrum who's 43 years old, they're a non-smoker, they have really good A1C, and they have erectile dysfunction. And I think we all look for unexplained variation as where we're trying to understand a problem, and that's where the opportunity is to ask questions. So that's just sort of a snapshot of the population we're talking about. And the ultimate vision is that we could come up with some mechanistic basis for specific types of ED. And this is adapted from a great article in Lancet by a asthma researcher, but I liked it. You know, we have the exposome, which for my study this is glucose, this is reactive oxygen species, this is cigarette smoking. Those are some of the big things that are causing ED. And then we have genomics. And I mean, what I'd ultimately like to have is a multi-level study where I had DNA, RNA, and the inflammasome. You know, unfortunately, in ED, we're not going to get corporal tissue for RNA sequencing. That's just not going to happen. But we can get DNA and we can get other measures. And presumably there are different mechanisms going on relating to smooth muscle or endothelium or nerves, and that they're leading to these phenotypes. So we still haven't discovered these endotypes, but we believe they're probably present. And I'm going to show you some examples that maybe are getting at this from a different perspective. And this was the game changer. So one of our fellows who was supported by our T32 took all the data from the entire time span of DCCT EDIC. And every year the patient was asked the question, were they impotent, yes or no? And this heat map, every row here is a patient of the 600 and some patients in the study. And every column is a year of the study. And so what you can see is up here, this is the big group of patients who never ever reported impotence. So they always said they were potent. And you can see this gap here is between DCCT and EDIC when we had a year gap in the data, but they're all blue. And down here is another group, and this is the patients who, once they develop ED, they have it for the rest of time. And quite honestly, before we did this work, I felt like this is probably the way ED is, that it's this binary thing, a point of no return. You get ED and then you have it, like a cancer diagnosis or something. But look at this. It turns out that the vast majority of people are in these other sections. There are 250 who have this intermittent ED. And so they might have a period of ED and then, then no ED for a year, and then more, and then no, and then they go to yellow. And some of the people here change status up to 7 times. And so I'm going to surmise that this group of intermittent patients has a different endotype, like a different underlying mechanism of ED than the one at the bottom. Our hypothesis is that maybe this is a neurogenic problem down here where they don't return. They hit the point of no return and they can't get back. But this is a really interesting group here. I think my next slide just zooms in a little bit on it so you can see that we call these the flip-floppers because they're flip-flopping in and out of a state of ED over time. And the hypothesis is that maybe there's a different set of mechanisms going on, and that these are people who might be subject to an intervention study, because maybe we can turn them back. You know, if they're still in this intermediate state where they're not profoundly dysfunctional, we might be able to reverse the tide. So we did some preliminary studies looking at what predicts remission, because so some people remit, they have ED and then they don't. And the duration wouldn't surprise you. The longer you've had the stretch of ED, the less likely you are to go back to a potent state. And body mass index was associated with a lower chance of remission. Then so was hemoglobin A1C. You know, the higher your A1C, the lower the chance of remission. And things like blood pressure and smoking didn't in our analyses. And this is just a hypothesis, and it's hard to do further work on this cohort because they're so advanced in age and things like that. And we actually can't do any interventions in this ancillary study. It's just an observational cohort. But this is really where it started changing my thinking about how we think about ED. Instead of this one point, once you've got it, you have it. Maybe that's not the case. And since this is a lot about urinary symptoms and your group's really interested in that, I want to share with you that a similar phenomenon is happening in women who have urinary incontinence. We use the Sandvik scale to measure incontinence. It's a good questionnaire based, and you can see that there a cohort of or subset of women who have the intermittent type of urinary incontinence, where some years they have it and others they don't, and a persistent phase. And you can see that the prevalence of the incontinence is more like a third as opposed to a half that we're seeing here for the men. And it really got to this issue of the time scale is the thing that was missing from so many studies about benign urologic conditions, because you don't have the money to do NHANES every year, or you don't have the stand-up cohort stood up to study this problem. So this is just another way that we've been thinking about the, and I put this in here because these are urinary symptoms. This is urinary incontinence in women and AUA symptom scores in men. This is more of a clustering exercise when we have multiple years of a questionnaire. So these are not binary. So we have a continuous variable for a score here for incontinence in women and the AUA symptom index in men. But over time, there are people who vary in their symptoms, and Aruna and her team did some clustering exercises. You can see there are groups like this red line of people who have very low symptoms that are very stable. And then there's an intermediate group that clusters around a middle range. And then there's another that seems to be getting worse over time and is more severe. And I'm not going to get into all the details, but if you have a phenotype and you have multiple years, you can learn a lot more about the patients. And Aruna's vision is sort of that we could take different elements that go into this. So not just the diabetes variables, but things like health-related quality of life, depression scores, and this is basically machine learning to try and create different clusters. And this is a hypothetical drawing here, but I think this may help us with all the data that we're getting over many years with the repeated measures. I believe that we'll be able to identify, we're going to try and cluster the urinary symptoms and see if we can, and then use some of that multi-level biological information to understand the mechanisms. It's easier for urinary symptoms because we can get RNA from the urine and things like that. So take-home message 2 is don't think of benign diseases as irreversible. And, you know, a lot of the categories we make between mild, moderate, or severe LUTs, you know, if you're 1 point on the other side of the scale, you're going to be put in this category. So some of those categories are a little bit arbitrary. And so slight changes could lead to recategorization. So I think we have to be careful about what we're calling remission. I'll acknowledge that. And then, I mean, the last thing is just, there's so much data that we're collecting, and oftentimes we use so little of it. And so by having that heat map, I'm using 30 years of data on over 1,000 patients, you know, that's 30,000 bits of information. So I think we're just scratching the surface in what we're doing with the heat map. So I want to show you how we then, some of this experience we had with ED phenotyping, I want to show you how we pivoted to try and address some other important questions in the field of erectile dysfunction. Because about a lot of erectile dysfunction variation is unexplained. You know, you put in the blood pressure and the glucose and the age and the cigarette smoking and a couple other things, and you still have much unexplained variation. And it turns out a twin study from back in the early 2000s showed that about a third of ED is felt to be due to heritable factors. And it also turns out that the senior investigator was an epidemiologist at the University of Washington. I don't have a take-home message on this, but you know, make sure you check where the authors are from when you're reading a great paper, because I've had a couple instances where I just found by chance that they were at my institution and that I could just go down the street and talk to one of these people. And my conversation with Jack Goldberg back in 2005, I think, catalyzed something that I'm still working on today. So we wanted to study this, and fortunately, there was someone at the University of Washington, Gail Jarvik, who's a leader in this network using electronic medical records and genetics data to study problems. And again, I had a fellow who had time, which I didn't have, and he spent a year working in Gail's lab, and that's how this whole project came to fruition that I'm going to describe to you. And so what basically we realized we needed to phenotype differently to get the number of participants required for genetic studies. So DCCT EDIC had 600 men. Well, we need like 20,000 men if we're going to do a full-scale genetic study, or 30. So we were never going to do it with an existing cohort. So that's where this network came in. Can we apply their methods to a different problem and study ED? So we developed an electronic medical record algorithm for ED. And we started out with the assumption that we'll just work with older men who are more likely to have ED, and I'll come back to that. If they were younger, we excluded them. Then we excluded people who had radical pelvic surgery or radiation or prostate cancer, because their ED is going to be non-genetic in origin, and we excluded them. And then we used ICD-9 codes for an ED diagnosis, and CPT codes or medication from pharmacy to measure treatment. And then in this particular thing, we had some natural language processing, where if there were more than 2 or more mentions of ED in the chart, we considered them cases. And if they didn't have any of those, they were controls. We did make a decision that if they were older than 75, we'd exclude them, because there are many reasons why people may not be seeking care in that age range. We ended up with about 37% cases, 60% controls. I'm going to get pretty granular in this next slide, and I hope you can see it. This is just a chart. These are the exclusions in red. You can see the frequency of the exclusions was very low. There are about 1,500 people in this test sample. And these were the controls. They never had any diagnosis of ED, no procedures, no prescriptions, and a few of them had a single mention of ED in natural language processing, but very few. And then in the controls, you can see that the bulk of the controls had a diagnosis code, but a few only had prescriptions. And then a very small number had no diagnosis code, no procedure code, no prescription. So just natural language processing. So we were able to, we think, separate with our algorithm pretty well into cases and controls. We then applied this to a very large cohort through Kaiser of Northern California, where we had like 35,000 patients. And what you can see is, and in that case, we had that 4-item question. Amazingly, someone had the foresight many years ago to administer this single 4-item ED question to like a huge cohort that were also being genotyped. So most of the controls through the health record added up pretty nicely, but there were some cases in the health record through diagnosis or prescription who on their survey, they said they did not have ED. And it's interesting that maybe these are people who are actually being successfully treated for their ED. So when they say, do they have a problem getting and keeping an erection? If you don't ask them about their medications, well, they may say, I don't have a problem. The bigger problem is people who we classified as controls through the health record. So no diagnosis, no procedure. But they, in the survey, they say they're a case. They say they cannot obtain or maintain an erection. And it's a significant number of people. Fortunately, you know, we have so many other controls here that probably you can get around this in genetic studies just with the size of your control group. But that's a little bit of a problem. So But regardless of that misclassification issue, we actually found a hit here where a single locus with multiple non-coding variants near the SIM1 gene on chromosome 6 were highly associated with erectile dysfunction. You can see the p-value is 10 to the minus 25, so far above genome-wide significance down here. I want to share with you, we then did a couple other things. We looked at the survey that had the 4-item question, and you can see that someone who never or sometimes could get an erection, so this is more severe classification of ED, had a higher odds ratio of association with the gene. So people with more severe ED had a higher association with this particular variant. And then on the next slide, I'll just show you the other thing is that there seemed to be some age gradient where the younger patients were more likely to have this SIM1 association. So that would make sense. If you have a genetic cause of ED, it might have an earlier age of onset. And so we were excited about this finding, and I'll share just 2 slides about the mechanism, and then I'll wrap up with what we're doing currently for our phenotyping. So this was replicated in another cohort, of course, and what we did is looked at, well, where is this variant? And the variant is near the gene called SIM1, and it's actually in an enhancer region that through chromatin studies showed that it would interact with the SIM1 gene, which is actually itself a transcription factor. So it's plausible that if you had coding variants or something that affected, if you had some variation in the effect of this enhancer, it could have an effect on the promoter in SIM1. And just to close the loop on biological plausibility, so SIM1 is actually found in neurons in the hypothalamus. And this is a little diagram of a hypothalamic neuron that has a receptor for melanocortins and leads to penile erections in humans and in animals. And so there's actually a plausible pathway whereby disruptions in this gene could impair the central control of penile erection. And that's the thought whereby we might be seeing the effects of SIM1. So this is just my take-home message number 3. In the course of this event, we had to collaborate with a lot of people, people at eMERGE, people at Kaiser, people at UCSF to do the studies that you just saw. And I've just found that almost everyone's willing to collaborate, or they say they will. And then if they don't, well, you find someone else. But if they're excited about the science, they'll probably collaborate. And in the course of this project, I had other collaborators that we ended up not using at all because we just the project didn't fit in with what we said we were going to do. So we had to change plans, and we didn't complete all the experiments we said we were going to do. We had to keep moving with where the scent was leading us. So how can we advance ED genetics? One thing is discover rare variants that provide greater mechanistic understanding of ED. But I think what I'm going to focus on mainly is better phenotyping and bigger cohorts. And this is why I have this material here, because the last part of this talk is really about better phenotyping. And what our approach is to try and use that time span. As I was showing you in UroEDIC, we have 30 years of data, and it allows us to see that there are different subphenotypes of ED. The flip-floppers on the one hand, and then those who have the irreversible on the other. And so the electronic health record gives you a great chance to use time, because people tend to stay in a health system like Kaiser or others for a fair bit of time. And so you can capture that through the electronic record. And then, of course, the electronic records allow you access to bigger cohorts. Just an example about the rare variants, though. So this is a Kaplan-Meier curve for the development of erectile dysfunction. And you can see here, what I'm showing you is that if you have extra copies of the SIM1 variant, that causes the association with ED, you have an earlier onset of ED. You can see if you have one or two alleles of the minor allele here, you have age of onset is earlier. You can see we're going back to the mid-20s for the onset. This is based on the eMERGE data. So if you have 0 copies of the disease variant, you have this likelihood of ED, and it's shifted to the left earlier. So there's earlier onset. And these time to event phenotyping gives you more power for your genetic analyses. So then we actually did something interesting. We looked at rare variants, and we created a rare variant burden, and we did the similar thing. Now, in this case, what's interesting is the rare variant actually appears to be protective. So if you have 2 copies of the rare variant, you're out here and you have a delay in time to onset of ED compared to someone with 0 copies of that rare variant. So this would suggest that some of the rare variants are actually protective. And just one more slide about this. So it turns out that the rare variants will put you in one of 2 directions. There's some rare variants on chromosome 6 in the SIM1 coding region that will increase your risk of ED, and some will decrease the risk of ED. So we almost saw this cancel things out. So it looks like you're not seeing anything, but with a large enough sample and using our time to event, we can tease this out a little bit further. What about better phenotyping? So this is some work we've been doing at Geisinger, who's one of the eMERGE sites and part of our genetics of ED study, and my collaborators are noted there. And so we said, why stop at age 45? Let's go back to age 20. Let's try and find the really young patients, because some of them still could have a genetic cause of ED. And so now we're talking about 89,000 persons, but we have to separate out and just go with the males. And again, we define cases through diagnosis codes, procedure codes, medications. And then we wanted to know whether the number of encounters in the record mattered. And you can see here, we said you either had to have 2+ encounters or 3+ encounters. It didn't really make a difference. We were able to identify people with ED. So here's just, you can just look at one side. We had 23,000 patients in this particular group. When we looked, the controls were the majority, and that makes sense because we're dealing with a younger group. So we're not going to get 30% with ED, it's going to be lower. Most of our cases had organic erectile dysfunction. Very few had psychogenic ED, only 48. So if we miss those, it's probably not a big deal. Basically, almost no one is getting a penile implant or other procedure like that. And then some people are identified by pharmacy alone, but it's still the minority. So this is showing that we're trying to scale up to larger cohorts and refine our phenotyping. And I just have 2 slides more, really. Where do we want to go with this? Well, this is a slide. This is sort of the next wave of my heat map. And this is a group of 1,000 patients with erectile dysfunction within a Kaiser Washington cohort. And you can see we're starting at age 20, and every dot represents some interaction in the health record around ED, either a prescription or a diagnosis code. So either a visit for ED or a prescription for ED. And every line is a person, And so you can see that people come in at various ages into the health record. This person's coming in at age 30, and they're having a lot of encounters in the health record. This person is coming in at a later age. And what I hope, and I'll show you this next slide, I hope is that if we zoom in here, this is a zoom in on the young patients. So maybe we can actually use some machine learning to quantify severity of ED by the number of times they're in the health record. So we can get away from just a binary thing. So we have the age of onset, which is really powerful, but can we then quantify their severity by the number of mentions of ED, the number of visits, the number of prescriptions, the number of refills, the total number of Viagra tablets that they consume. This is, again, getting into data. Now we're going to have hundreds of thousands of data points, and we'll need even more analytical tools to refine it. So this is this a phenotype? I think there's so much here. I think this is like just filled with, I don't know if it's data or metadata, but it's filled with information that we can extract and hopefully make our phenotyping even more accurate. Now, why is this important? Because we need to have bigger cohorts. And in eMERGE, we have 18,000. In MyCode, we have 23,000. But look what we have access to now, the Million Veteran Project. We expect we'll have about 720,000 men with electronic health records, including diagnosis codes, pharmacy data, and genetics. And then the United Kingdom Biobank has hundreds of thousands of patients, and they're just adding their outpatient records and pharmacy data to the biobank. So again, we can use our phenotyping algorithm to scale up by, you know, an order of 10 or 100 to discover more common variants, to study rare variants, and to continue our work. So how do we put it all together? This is kind of my conclusion slide. What do I hope I can do in 5 years? Well, I want to assemble a massive cohort of men in the electronic health record and identify those at highest risk for developing ED. So they're going to have diabetes, they may have blood pressure problems, but before they have ED. So look at the known risk factors, add in our genetic variants, maybe develop a polygenic risk score, and then it's to intervene. So can we use this to find a large population for some sort of controlled trial, maybe a suite of behavioral interventions to improve compliance with X, Y, and Z, you name it, versus usual care, and then remotely follow them with remote monitoring, like, you know, quarterly IIEF score delivered to a handheld device and giving them financial incentives for reporting and things like that. So that's sort of the holy grail, that we could use this to develop a large enough cohort that we could do an intervention study and try and reduce the burden of ED in young men. So I'll finish with some acknowledgements, and I'm sorry if this took a little longer than expected. We have a urology research outcome collaborative at UW that's been invaluable, people from other disciplines. My UroEDIC collaborators, it wouldn't have been possible without them, in particular, Arunas Sarma, my EG genetics collaborators, and then my collaborators from the Urinary Stone Disease Research Network. That's where I'm learning about behavioral interventions and these other remote recruiting that I hopefully can apply to a different population. And then acknowledging all my former students and fellows, in particular, Jim Hoteling, who made that connection with Gail Jarvik, Melody Palmer, who worked on those initial heat maps, and a current grad student, Tianren Lee, who's working with that Kaiser data, with that purple plot of all those millions of data points. So with that, Chris, Will, thank you for letting me share my work, and I'm happy to answer all the questions.
Well, thank you very much, Dr. Wessells. That was a stellar talk, and you've generated a lot of questions in the chats already, which I've taken note of. But before I get to those, and they hit on things like the phenotyping methodology, as well as what you expect might be some potential therapies and interventions. But before that, I'd like to invite Dr. Aruna Sarma and Dr. Doug Strand to each take a turn, Aruna from the epidemiological perspective and Doug from the basic science perspective, to sort of give some insight or some thoughts about your talk from those perspectives. Dr. Sarma, would you like to begin?
Sure. Hi, Hunter. I was asked by Christina to comment on sort of the epidemiology aspect of this. And of course, I'm biased because I think what we did was phenomenal and has a lot of impact. But I want to kind of describe why I think that's true. I think with epidemiology, there's a very sort of systematic evolution of how we conduct research to ultimately get to our aim, which is intervention and prevention. And most of the time in clinical research, we don't adhere to that timeline. We skip to trials and big cohort studies before understanding sort of the descriptive epidemiology. And one thing we did really well here, which I think is a really good example, is we started out in that contract really understanding burden and descriptive epidemiology. We tried to generate hypotheses about risk factors. And we did that well before we had the funds and the time to be able to look at this cohort periodically. And once we were able to do that well, we knew when we were going in with 10 years of study that was going to ensue exactly what kinds of risk factors to look for. We could understand incidence, so we had a sense of risk, as well as the types of factors that we could now test in terms of hypotheses, not just generating them, but we were able to test them. And of course, now with the 15 years with the great heat maps that Hunter provided, was really understanding natural history. And I think what comes next, which is where a lot of the methodologic stuff that he is talking about really becomes helpful, is the last stage in terms of sort of epidemiologic evolution is really screening and intervention. And we can't do that without an understanding of natural history. And so I think the next steps are really going to be, understanding where we can identify, screen, prevent, both primary, secondary, and tertiary. We can identify these people before they get ED, perhaps in that intermittent stage where most of the risk factors were associated with behavioral, risk factors and hemoglobin A1C, glycemic control. We can prevent some of the further damage that may happen, and then maybe see what we can do, before the lasting effects of the neurologic complications occur. So I think we've done this right when it comes to just the natural history of epidemiology. And it's a good example, I think, for other studies for sure.
Excellent.
I think my response to Runa would just be, being a urologist and working with an epidemiologist has been so valuable, because there's just some aspects that I understand from seeing the patients, and then you get motivated. Now when I see a man with an A1C of 9.5 and they're overweight and their exercise is not very high, I'm not thinking treatment. I'm also thinking, okay, let's get beyond just the treatment. Can you do these three things? And that's what's motivating my idea for a clinical trial, because we see it in practice. And of course, then I don't actually have to answer any epidemiology questions, because Aruna answers them for me.
And
Brilliant.
and I start talking to people about ED, which is never a good thing.
Dr. Strand, from a basic science perspective, what does this study tell us?
So, I think the most striking thing is just that we can't get tissue from this study. So how do you ever get to a mechanistic understanding of what's going on beyond the genetics of it? So this is why I was asking a question about imaging earlier, or something like that you can correlate function with the clinical phenotype. And I think you could probably start, my emphasis in the BPH world is to look at things at the single cell level to try to understand biology. And I'm wondering if that would be the next step here, is to look at these genetic risk variants and try to pinpoint specific cell types. You were saying neurons might be it. Maybe there's others that you could eventually figure out function, biological function from. But that's a really tough leap to make when you're not getting tissue from these patients. So, on the cell biology side of things, I think that's a big hurdle, and I don't know how you get past it. But otherwise, from a clinical side of things, Dr. Wessells, that was tremendous.
Well, I agree with you. The challenge of not having tissue is really major. One thing that we've been thinking about is GTEX, and unfortunately GTEX, it's a compendium of mRNA expression by organs, but they actually don't have any penile tissue in GTEX. So we proposed in a grant to create a GTEX-like library of normal expression, from cadaveric, donors, organ donors who are undergoing organ retrieval to obtain penile tissue from different ages, do the mRNA expression, and then you at least know what's expressed in the normal, and maybe use that as a way to make connections to other levels of data.
Absolutely.
And then I don't know if eventually blood would be a source of information around gene expression. That would be require just incredible amounts of sophisticated data analysis. Could you ever imagine that?
Absolutely, yeah.
That kind of dovetails into a question that Dr. Casey Stedman asked. She's a K-12 scholar at Duke. She is wondering, would there be any benefits to having quantifiable penile pressures to further delineate types of ED, like similar to the telemetric pressure transducers that are used in animal models, but less invasive in humans?
I mean, that gets to the idea of having some sort of quantitative measure to go along with the self-report data, because then you can do a lot more. And I mean, there are obviously Doppler studies that can be done to measure blood flow, nocturnal penile tumescence testing, which could be, those have really not been used for research purposes correlating with the phenotypes. They've just been used as a clinical tool. I think we're missing something like that. I quite honestly, I don't have a good answer to that. This is one of the challenges.
Well, I see an entree for Dr. Stedman to maybe get involved here. Stephen Vandeneden is wondering, what is the impact of using bladder function, like as measured by IPSS or AUASI, in trying to phenotype erectile dysfunction?
Well, I mean, we know that bladder, there is this association between ED and lower urinary tract symptoms, and it's unexplained really. So I think it has to be an important, probably covariate. But if Stephen's question is, could you use bladder tissue as a surrogate for erectile tissue? I think the sort of the neural control and the functions of it are different enough. That would probably be tough. Doug could maybe think more about that, but I don't think it would work.
Please remember to fill out the post-event survey. Catherine has put another link in the chat box. There's a question from Greg Whittemore about those flip-floppers that you were talking about. What sorts of interventions do you envision? Are treatment modifiable vascular risk factors, or do you see potential for like shockwave therapy?
Well, I think it depends maybe where they are. If they're early on, I think we'd want to try those modifiable things. But the shockwave is a really interesting thought, because it's thought to enhance, vascular function. And I guess what I'm hoping is that we can identify a vascular phenotype and maybe a neural one, and then appropriately treat. The shock wave might be beneficial for a vascular phenotype, but not a neural one. And medications is obviously another one, but I guess I'm trying to move earlier than medications.
I'm wondering about patient engagement and patient involvement. What have you experienced, or what have you done in terms of that, and what have you learned?
Well, there was a lot of concern that people would be worried about privacy and not want to fill out the questionnaires, but we were pleasantly surprised that, over 90% of the participants in UroEDIC agreed to fill out the questionnaires, and there's almost no missing data. So I think the fear of that is over emphasized by IRBs and things like that. And people are concerned about this as they age. So I think being upfront and informing them about it, they wanted to get the information back. And what else can we do about patient engagement? Well, that's some of the things. That's why I mentioned my Stone Disease Research Network, because we're doing an intervention study to enhance fluid intake to prevent kidney stones. And so we're learning a lot about that I think we could apply to other conditions, behavioral interventions and retention in trials. It's tough. These people are really invested. And so how do you get the similar level of investment? It's tough.
Laura Pascal notes that it might be interesting to see how the pandemic impacted erectile dysfunction, and because there are many cases that are probably exacerbated by the whole pandemic. What do you think about that?
I think there have been reports of that, and it's unclear whether there's some sort of vascular inflammatory effect of COVID infection. And then you think of just pandemic, even on people who don't get COVID, you know, inactivity, dietary changes, you know, for all the people who are exercising more, there's a population that are probably falling further behind. I don't know what its effect on diabetes control is, but we're pretty sure there's going to be a long lasting impact. Yes, the resveratrol in the red wine might help. So maybe we should work on red burgundies.
There's an idea for an intervention, right? Well, Dr. Wessells, thank you so much for talking with us. Really, again, just a stellar talk. And I can see sort of a blueprint or a roadmap for how we might use what you've done and sort of the order that you did it and approach other urologic diseases. Any other comments, Dr. Sarma? Would you like to make a closing comment?
No, I just want to echo, working with Hunter has been phenomenal. I've learned a tremendous amount. I think UroEDIC should be an example to how to navigate these, you know, these large studies that NIDDK has already put a lot of resources into. We've done a really good job working with them and capitalizing on that strength. So
thank you.
Yeah,
Remember, go ahead, Dr. Wessells.
Just this is the tip of the iceberg. I mean, there's so many other benign conditions that I think this roadmap of getting some experience in a well-described cohort and then moving into the uncharted territories of electronic health records, and with MVP, the Million Veteran Program, and all of us, you know, there's just going to be huge resources for everyone to take advantage of in the coming years, so that we don't, even if we don't have cancer centers and cancer consortium grants to stand up these huge repositories, I think we can get access to the information we need.
Well, thank you very much. This talk has been recorded, and with Dr. Wessells' permission, we'll be putting it on our CAIRIBU YouTube channel. And so you can tell your colleagues that might have missed this to go there. So we'll send out a link for that. So thank you again, and thank you for everybody for participating.
And if people have questions for me, I kind of went over, send an email, wessells@uw.edu. I'm happy to sort of have a chat on sideline as well. So thank you for the opportunity to inaugurate this Catalyst program. It was really fun, and I'll look forward to future meetings.
Thank you so much. Bye-bye, everybody.
All right.